yesterday I was doing medical students, and then immediately after me was Dr. Eldridge. And Dr. Eldridge and I were kind of preoccupied with other people in another building. So we are very happy that you're willing to change a Thursday class to a Friday class, and I really appreciate it. It's good to be able to be versatile like that. So today we're going to start what would be 3 lectures on metabolism, and the first one will be our growth hormone. Then we'll move right into the thyroid hormone regulation. And the thing that both growth hormone and thyroid hormone have in common is that they are very key towards growth in phases of development where you need to have growth, but their normal maintenance in adult is largely going to be involved in metabolism and energy. So then after that we'll move right on into a general overview that includes the pancreatic hormones, insulin, and glucagon, which are the ones you probably think of when you think about energy regulation and uptake after food. But we'll start here with some global pictures. So last time we talked in this room, anyway, we discussed all of the pituitary hormones, and growth hormone is one of those, and how most of these were made as peptides cleaved into smaller peptides if they needed to be. And growth hormone was one of those larger hormones that was very similar to prolactin. So it's a larger peptide hormone, not a protein, but a peptide. And then we discussed that growth hormone actually has 2 functions, and that the growth function is largely mediated by a compound called somatomedin, and the other name for it is IGF, which is related to insulin-like growth factor. So the reason that this was discovered and the reason that it has 2 names is that different people in research labs are studying different things. So the folks who were actually studying growth hormone realized that since growth hormone could be identified and purified from the pituitary, or even just provided as a pituitary extract, if you injected it in an animal model, you could see growth of tissues. So you could see growth of the muscle, you could see effects in the skeletal system. But if you took these individual tissues out and put them in a water bath where you could be bathing them in isotonic saline with your pituitary extract or your growth hormone, you couldn't reproduce those effects. And so they began to start searching for something that could mediate those effects. And they found that when growth hormone was given to the liver would start making something, and they called it somatomedin until they found out, well, otherwise. So, you know, if you're the first one to discover something, you actually can name it, and they named it somatomedin. And what they discovered was that whatever this was, and we now know it's a cytokine, came from being stimulated by growth hormone in the liver, and then this somatomedin was able to interface with the skeletal system, interface with muscle for growth, and actually, as we'll see, it'll feed back and regulate the further release of growth hormone in a long feedback mechanism. So they named it somatomedin and began studying how somatomedin worked. Now, meanwhile, there were people that, particularly in using cell culture, were studying how insulin works. And insulin also causes growth. And insulin generally has its effects you think of as mediating the uptake of glucose into tissues right after a high glucose meal. That's what you think of when you think of insulin. But insulin has other effects that are largely related to growth, and particularly if you give a larger concentration than what you needed for glucose uptake. So they reasoned that maybe it wasn't insulin working through the insulin receptor, maybe it was something that was insulin-like. And maybe it was working through the insulin receptor, but they didn't know. So that lab started looking for something that they named an insulin-like growth factor, because they thought that insulin's effects might have been mediated by something that wasn't like insulin but behaved like insulin. So they named it insulin-like growth factor. And it took a while to figure out that these two factors were exactly the same thing. So now in our literature we see somatomedin, we see insulin-like growth factor, IGF as its abbreviation, and it is the same thing. So what does it do? It causes growth in a cell culture system or in an animal model or in tissues. It increases amino acid uptake. It also uses those amino acids for making new proteins. It increases RNA and DNA synthesis so you can get proliferation. And insulin is a mitogen, and insulin-like growth factor is a mitogen. So that means that it causes cell division as part of the growth. And then increases collagen levels in bone and skin, increases not only cell number, but also increases cell size. So this was what was mediating the growth.
So are they the growth hormone and IGF are the same thing?
No. Growth hormone induces the production of IGF in the liver.
But they act on the same things?
Yes, yes. So if you gave growth hormone, then your liver would be the target, and the liver would then make the somatomedin, or the IGF, and that would be released into the body to produce all of these effects.
So why does it show like the arrows coming from growth hormone to like the same?
Because it also has effects there by itself. So growth hormone actually can work at the osteoblasts, and it can work, and I'll show you a figure on this in just a minute, as a paracrine regulator to induce IGF levels in the areas where collagen needs to be made. So it can work there. Now growth hormone can directly have effects, and particularly here in adipocytes, because that's where it's having its metabolism effect, to be able to release lipids so that they can get into the bloodstream to be used for energy. So that's the second main effect that we think of as growth hormone effect. So this is how that works. The pituitary releases the growth hormone. The growth hormone actually has a binding protein that carries it, not because it isn't water soluble and needs to have something to carry it, but because by having this protein that binds to the growth hormone peptide, it allows it to be a storage site in the blood supply, so that even though you may have one bolus release of growth hormone here, it can maintain growth hormone activities for a longer period of time by having that protein that it can bind to. So then the regulation is that when your growth hormone is released from its binding protein, it goes to the liver, and it signals the production of IGF. So IGF can be synthesized in the liver. And the reason this says target organ is because there are other targets in the body, such as the bone, that are going to also make IGF in response to growth hormone. So then IGF is made and released from, let's just say, the liver, and then it has a binding protein as well that regulates its storage as a plasma protein, and then it acts as a cytokine receptor. So this insulin-like growth factor works at a tyrosine kinase receptor that looks a lot like the insulin receptor. So it's the insulin-like growth factor receptor, but it looks a lot like the insulin receptor. So it's a tyrosine kinase receptor. This is a cytokine receptor, which is a tyrosine kinase-like receptor. So it goes through those JAK-STAT pathways to get signal transduction through the nucleus so that we get new protein synthesis that can cause growth. So IGF, or somatomedin, is one of two forms. The one that we call IGF-1, or somatomedin C, that we have largely in adulthood. So that's what is now mediating the effects of growth and proliferation that you have to maintain your bones. And then we have IGF-2, which is the fetal development hormone. So that's somatomedin A. And then after the baby is born, there's a period of time when the baby has both IGF-2 and IGF-1, so that both of those are involved in infant growth and development. Now this is a larger protein, 7 kilodaltons, I think could be called a protein. It's fairly small for a protein. It probably won't show up very well on a SDS page unless you provide an environment where you can maintain very, very small proteins. So it's a very small protein, but it does resemble proinsulin. So insulin, we'll see in the next time I meet with you, is a large prohormone that is cleaved after it is circularized and it finds some secondary structure by sulfhydryl linkages. But before it's cleaved, that would be the proinsulin. And that's what our insulin-like growth hormone looks like. So growth hormone's actions at the skeleton and for visceral growth are mostly mediated by IGF. And it's largely attributing IGF, not growth hormone itself, for the actions that are cell proliferation, the differentiation of those cells after the mitogenic activity has increased the number of cells, as well as the new protein synthesis in other cells. Yeah?
What happens if you give an adult IGF-2?
Pretty much the same thing.
Okay.
They resemble each other. I think they're all going to be working on the same receptor.
Okay.
So I'm not exactly sure why placenta would make one form and not the other. But then I can't tell you why placenta makes chorionic gonadotrophin rather than luteinizing hormone either. I can guess there are things that the baby needs, or the fetus needs when it's developing that the mother doesn't want. And likewise, there are hormones that the mother wants that the baby doesn't want. And so your fetus begins developing its own endocrine organs fairly early on, sometime between first trimester and second trimester, where it can begin maintaining its own thyroid glands and its own other glands. And so it's possible that maybe this is part of the situation, so that mother's hormones are not going to be largely influencing the fetus in its own development.
Cool.
So the fetus has its own regulation of its own development. And the circulation is different. The mother's circulation has to abut against the fetal and placental circulation in order to have things cross. So it's not like mother's blood is going directly to the baby. That won't happen. Okay, so here's a depiction in another form. Here's our growth hormone. Our target tissue, such as the liver, is going to be causing an endocrine release of IGF, because here's the target, the receptors are here at the liver, the IGF is released into the bloodstream, so now it becomes an endocrine hormone. But on the other hand, if our growth hormone is working at tissues where cartilage is being made and the bone, then it is not getting into the bloodstream, but it is regulating the cells in those very same tissue. So that is called a paracrine effect, when one cell type in a tissue is communicating with its neighboring cells by releasing something. So in this case, the IGF doesn't ever need to get into the blood supply. It is communicating right here. And it's increasing proteases that are necessary to cleave from the binding protein that's in the bloodstream. And it's also in this tissue stimulated by parathyroid hormone and estrogen. So they can work through the osteoblasts and just as well as growth hormone can. So here in the bone is where IGF, under the influence of growth hormone, is having many of its effects to maintain cartilage and long bone growth in development, and then maintain the bones in adulthood after long bone growth has been stopped. So our regulation here is that once that IGF has been released, it is transported by an IGF binding protein that maintains a pool in the blood supply. And then when that needs to be released, it triggers a protease that can cleave this binding protein off, and then you have the IGF. So the IGF is largely taken up in the liver by this insulin-like receptor. So this is a tyrosine kinase receptor. And in general, insulin-like growth factor, all of these growth factor receptors are internalized into endosomes, into the cells. And that's the main way in which the hormones themselves can be degraded, because they bind to the receptor gets phosphorylated on tyrosines, it gets internalized in endosomes, it brings the insulin or the insulin-like growth factor in with it, and then they get degraded. So a large supply of something in the liver is going to be degraded by those receptors right here in the liver. And since this is a tyrosine kinase receptor, you can see that it can work through AKT and the protein kinase, or same name, protein kinase B is AKT, to regulate some of those effects that other tyrosine kinases can do. So those effects can include sugar and amino acid uptake, protein and DNA synthesis, mitogenic activities, and for osteoblasts, that's very important because the osteoblasts are building the bone. And after they built the bone, then their half-life is only a month or so, 2, maybe 3 at the best. So you need to be continually building your osteoblast population. And as you recall from the day before yesterday, the osteoblasts are controlling all of that bone regeneration that occurs. You're rebuilding your entire skeleton every 10 years. So you need to have this going on continually as a bone regrowth mechanism. So this is really very important for that. Okay, now if we want to look at prenatal growth, we've got placental lactogen that works just like the growth hormone. And you might think lactogen sounds a lot like prolactin. You would think that would have an effect, but the placenta is not providing prolactin-like compounds for breast development of the mother. See, those circulations are different. They've got different endocrine systems, and they're working individually, the mother and the fetus. So the placental lactogen is the hormone that was named probably because it looked like prolactin, but it doesn't act like prolactin. It acts like growth hormone, and it's very homologous to growth hormone. And then our insulin-like growth factor 2, and that can be regulated by, instead of growth hormone, it's regulated by the placental lactogen. And the little H in front of proteins and enzymes means that it's the human form. And that's important because many of our hormones that we need to supply for purposes in treatments comes from isolated tissues from people. And the placenta is an extremely good source of tissue for gathering of hormones. Because, first of all, it makes so many that might be useful. So human placental lactogen and other hormones could be coming from the placenta. And then the other hormones that are growth-like that can come from the placenta are chorionic thyrotropin, and then the placenta can also make our estrogens and progestins and all of these can be involved in prenatal growth regulation. Then once the baby is born, we continue to utilize growth hormone, and at that point we have some IGF-1, which is the adult form, and IGF-2. And postnatal growth is also largely under direction of thyroid hormones, estrogens, androgens, as the case may be, which tend to be released a little bit immediately after birth, and then are very low levels right up until puberty. But right after birth, you tend to see these as continuing to be growth factors. Now, if you have too much of something, that can be detrimental as well, or too little. So too much glucocorticoid is going to be damaging to growth, and too little thyroid hormone can result in growth retardation as well. And we'll see an example of that later on today. So then we have a long period of time until we wait for puberty, and there growth hormone is very important again. IGF-1 under the stimulation of both growth hormone and insulin's induction. And there we begin to see where our gonads and our secondary sex characteristics are going to be involved in producing and utilizing estrogen and androgens respectively. Okay, so I just wanted to point that out, that growth hormone is not the be-all and the end-all of growth, that there are many other hormones that all are contributing. So let's look at our feedback effects. So we talked last time about the kinds of neuropeptides and stimuli that can release growth hormone, and that's going to be largely through growth hormone releasing hormone and blocked largely through somatostatin. And so they're receiving different kinds of regulatory inputs to ultimately modulate growth hormone release from the pituitary. So when we look more towards growth hormone as a growth factor going through IGF, IGF can feed back to the cells that are in the hypothalamus that are going to be releasing somatostatin, and the pituitary releasing somatostatin, and it can stimulate somatostatin release, which is going to be blocking growth hormone release. So that's a positive feedback to the cells making somatostatin, but somatostatin has a negative effect on growth hormone release. Now, we haven't really talked too much about this, but we will in just a minute. The other effect that growth hormone has is at the fat cells. And the fat cells, in response to growth hormone, are going to be releasing free fatty acids into the bloodstream so that they can be used for energy. And so those free fatty acids are going to be returning to stimulate a negative feedback here at those cells releasing growth hormone, releasing hormone. So the negative feedback is here at the hypothalamus. Now you might ask a little bit about how that happens. I'm very interested in growth and signaling due to G protein coupled receptors, and when the human genome was identified, you could tell what a G protein coupled receptor was because it had 7 transmembrane helices. And those helices are largely hydrophobic amino acids so that they can form a helix that's going to be inside the lipid barrier. So if you can identify patches that are about 20 amino acids long that are all mostly hydrophobic, you can say, aha, that's going to be a helix that can be embedded in a membrane. And if you see 7 of those in a row with little loops in between, aha, you've identified 7 transmembrane receptors. But that doesn't really tell you what they do or what they are. So in the Human Genome Project, they just started numbering those. And a whole series of them now have been discovered to respond to fatty acids. Short, saturated fatty acids, mid-range fatty acids that are saturated, unsaturated long fatty acids, so that tissues in the body can actually recognize when fatty acids have been released. And that's here, probably exactly what is signaling that fatty acids have been released is that there would be cells that can be detecting these because we've got a receptor for them that we know now. Okay, now growth hormones' effects and their feedback are largely going to be related in adults to the energy levels that we currently have. So the two effects that growth hormone have, we just discussed what we can call the indirect effects, are the insulin-like effects. And so if someone says to you growth hormone has insulin-like effects, it's because it's working through IGF-1. And the kinds of things that growth hormone has in common with insulin is the anabolic effects. Now, anabolic means that you're in a fed state, you've got nutrients so that you can begin utilizing those immediately for energy, and you don't need to break down any stores. You need to be at a stage where you can store some of those nutrients that are coming in. So at this point, insulin and growth hormone both lead to increased uptake of sugars, uptake of amino acids, and so another way of saying that is nitrogen retention. And then they lead to the protein synthesis and long bone growth. So insulin and growth hormone have these kinds of things in common. On the other hand, the direct effects that growth hormone has on its target tissues, which can include bone and several other tissues, is often going to be an anti-insulin effect. So here at the adipocytes, insulin's job after a high glucose meal is to be able to take that glucose into cells, including cells that are adipocytes, and into cells that are in the liver. And the cells in the liver are going to be making fatty acids out of those excess glucose molecules. So insulin's job is an anabolic effect to take those up, whereas growth hormone's effects is to increase lipolysis. So here we have what would be an anti-insulin effect, because the growth hormone actually decreases glucose uptake and decreases fatty acids going into the adipocytes where they can be stored. So no matter what you say about comparing growth hormone and insulin, you really have to look at it in context. You can't just memorize growth hormone has an insulin-like effect. You have to know what that effect is. And here, growth hormone has an anti-insulin effect. And some people will say growth hormone is anti-insulin cortisol-like because cortisol also releases lipids from the stores and prevents the lipid uptake into where it can be stored in adipocytes. So you have to be really very careful before you start memorizing things. You have to really think about what context they're in and what you really mean by anti-insulin. So we have a situation here where growth hormone needs to have energy being supplied, and the best energy it can supply is to release those fatty acids, and we call that mobilizing fats. And of course, all of that is going to depend on what your nutritional status is. So you can't just memorize it's going to mobilize fats. You have to understand a little bit about where you are in your state of healthy eating and non-healthy eating. So you've got, once you have had a meal, as the glucose levels rise from your meal in the blood supply, or the amino acids, and typically if you're going to be using this as a test model, arginine is used. And when arginine is supplied, you get a release of insulin from the pancreas. And then very shortly after that, with these amino acids in the bloodstream, you begin to see a release of growth hormone. So the insulin and the growth hormone are not occurring as peaks at the same time. The insulin has taken the glucose So when we have increased protein intake in a high protein meal, then we're likely to have an increase in both insulin and growth hormone. Now when we have our increase in growth hormone, that's going to increase IGF, and that's going to allow new protein synthesis. And the same thing is going to happen with that insulin. Our insulin's goal is to bring in storage, and so it's going to be bringing in glucose. The IGF-1 is going to be helping to bring in the amino acids, and you can have increased protein synthesis in fats and muscle. Now, if you have a high carbohydrate meal, then you have increased insulin, but the high glucose levels in the bloodstream decrease growth hormone release. And if you're a memorizer, you remember from last time, low carbohydrate levels was one of those things that could allow growth hormone release. Hence the opposite here, increased carbohydrates are going to decrease growth hormone release. So now you have, under a high carbohydrate meal, and we can pass around those high sugars right there. Anybody's ready to try this again?
Might as well split that one sugar.
You are going to be suppressing your growth hormone release. So that means, you know, you may or may not be having those stores of IGF-1 being involved, but now the dominant effect is going to be the increased insulin, which is going to cause fats to be stored in the adipocytes. So this is a coordinated effect, and its goal here is to store fuel. So storing fuel in the liver, storing fatty acids that the liver makes into the adipocytes, it's a storage. And so that's consistent with decreasing the utilization. Yeah.
Does the IGF-1 has a direct effect on adipocytes? Because in the previous graphs, it doesn't have an indirect effect.
The growth hormone's direct effects are on adipocytes,
But not through the IGF.
not through the IGF. Yes. Okay, now if we're fasting or under high exercise or any other stressors, because growth hormone can be called a stress hormone. We tend to think of glucocorticoids and epinephrine as being our most dominant stress hormones, but here under this situation we have decreased carbohydrates, so that increases growth hormone levels, but it also at the same time decreases insulin levels. So under these conditions we're going to have increased growth hormone. That's going to be causing lipolysis as a direct pathway. Now, under fasting conditions, we tend to have decreased IGF produced. So the IGF effects are not what's going to be dominant. It's going to be the release of the fatty acids so that they can be used for energy. Yeah, we have a disease that you're probably never going to see here in the United States, but if you're going to be traveling in places where there is a lot of nutritional deprivation of proteins, not necessarily carbohydrates, but protein deprivation. There's a disease called kwashiorkor, and without those proteins, you cannot have any building blocks, so there's very stunted growth during that time. But when you look at growth hormone levels in the bloodstream, they are extremely high compared with normal protein. And if you just give carbohydrates, that doesn't affect this at all. On the other hand, if you give protein, the growth hormone levels begin to normalize and are back at normal by 25 days. So when you stop to think about this a little bit, you've got to figure what is taking place here. Well, you can be providing lots and lots of stimulus to get an effect taking place. But if you don't have the amino acids to be able to take cells so that they can make proteins, then all the growth hormone in the world is not going to be stimulating those effects. And so you're just simply not going to have that happen. And one of the modulators that you remember that modulates the ability of translation of proteins is the mTOR pathway. And one of the regulators of the mTOR pathway is amino acids themselves. So if you're missing some of the key amino acids, then you just shut down translation altogether. So you can have very stunted growth under conditions of extremely high growth hormone. And then once you've supplied those key amino acids, then you can begin getting translation going on again, and you can be building in the tissues that need to be built. Okay, so if we have growth hormone deficiency in childhood, then those long bones just don't progress and get built. You have short stature. But if you have it in adults, then you see decreased immune functions. And that's probably because IGF is a cytokine. And so it has potentially other effects that may be involved in wellness in the body. Decreased bone mass, so that means your bone mineral density, and a decrease in lean body mass. So your lean body mass, that's mostly your skeletal muscles. So you can have skeletal muscle weakness. Now, not everything is due to growth hormone deficiency that we can associate with stunted growth. There are some people that may have a decrease in growth hormone receptors, and if you have a decrease in growth hormone receptors, then you can have lots of growth hormone, but you won't have any IGF made, because you need the growth hormone receptors to have that induction process. So the way to test this, and the name of this disease is called Laron dwarf, is that you just won't see growth hormone IGF production in the serum. And then there are problems in cartilage-forming cells in which they cannot respond to IGF. So just don't jump to the conclusion that all growth problems are related directly to the growth hormone in the body. So if we have growth hormone during childhood, then those long bones are going to be developing even longer, and you have what we call gigantism. If you have retarded growth, we just discussed that it may be due to a number of other things. And in fact, when we listed all of those hormones that are involved in postnatal growth and growth in puberty, you see that there are many other things that can cause problems. Now here's our growth excess in adults, and that's called acromegaly. And at this point, your long bones have, the epiphyseal closure has assured that you aren't going to get any lengthening of your bones. What you are is going to get thickening of your bones. And you can see this sort of thing. Here we have a young adult, and as she began getting to be older into her adulthood, you can see that jaw bones begin to be thicker, cartilage along in the nose is much thicker, and ultimately her skull shape has now changed a little bit. Hands are thickened, feet are thickened, and that's part of the effect of acromegaly.
Was this what Andre the Giant had, or was he just naturally a...
Well, he was,
Big
during
guy.
childhood, he was Andre the Giant because his long bones were growing. This is Abraham Lincoln.
Oh.
So Abraham Lincoln was believed to be acromegaly.
So he had both Marfan's and this?
Could be. I think probably we won't know exactly what he had, but some people will think it's acromegaly. So visual problems. Now, why might we have visual problems in someone who has growth hormone excess? Yes.
Yeah, it could be a tumor in the pituitary, which is near the optic chiasm.
That's right. Exactly. Does everybody agree with that?
I was going to say the same.
Okay, of course. I'll bet you all were gonna say the same thing, and you just didn't say it fast enough, right? So for some people who do have, you remember 50% of your cells in the pituitary were growth hormone somatotrophs. So if you're gonna have a pituitary tumor, it's a very high probability it'll be a growth hormone secreting tumor. So very often, pituitary tumors are diagnosed by people's ophthalmologists and optometrists because they go in, they've got visual problems, and that's where this can happen. So, and I guess the other thing is it could come from problems that might be from growth hormone releasing hormone, but more often than not it might be the pituitary. So here's another situation, and I'm going to leave this with you to take a look at, but I just want to show you this is current times, and there are some websites here that have talked to Tanya, cute little girl when she's 7, and then she went to college, and here she is on the beach in her bikini at 20 years old, but then she started gaining weight, and by the time she was 30, she looked like that. So she's got situations, and she didn't survive, actually. So
How tall was she?
she got to be 6'6".
20 and 30.
Because I was looking at the car.
Yes.
Hey.
She gained 350 pounds, yes.
If I had that height, I'd be an NBA right now.
Okay, now let's go on to thyroid hormone. So if you want to stand up and do a couple of stretch exercises, please do so right now while I pull this out. Okay, everybody stand. Take a deep breath. Stretch a little bit.
At least. No one really knows how many times you had it, but.
All right, to your shoulders. Actually, I'm a member of a bell choir. We do this all the time.
He is okay.
Yes.
It's like, oh, wow.
What are you doing? That's not in the stretches.
Okay, we're going to talk about now thyroid hormones. And thyroid hormones are very permissive. So you need thyroid hormones for growth. You need thyroid hormones for development. You need thyroid hormones to maintain your brain function. And I will tell you, I had a friend, and I don't know if I could really call him a friend, but I knew him fairly well because we sat next to each other on study sections. You know, he was a PhD. He had a certain level of brilliance that it takes to be at a PhD and sitting on study sections. And when I took my job at NCCU, it was to build a research program and in substance abuse, and so I wanted to hire him. And I phoned him up, and I never got any response. And then I got a response from his wife that said that he'd been ill and sorry. Well, what happened was he was hypothyroid, and he didn't realize it. And one day, his wife said to him that he was just behaving like a very stupid person. And she took him to the hospital, it was so bad. And she was really worried that there might be brain tumors or something causing his cognitive dysfunction. But she was scared to death. And he didn't necessarily realize he had this problem. But it was part of his hypothyroid disease that his brain just wasn't working. So after he got treated, things were fine. But for those of you interested in the central nervous system, the brain responses, even after your brain has been developed, can be under thyroid hormone So we're going to talk about what makes the hypothalamus work. And we have our releasing hormones from the hypothalamus. Now they can respond to a number of stressors. In animal models, we can see a lot of thyroid hormone releasing hormone if you put your animals in a cold room and they respond to cold. For most of us as adults, we use behavior modification when we are addressing cold, and we put clothes on and we stay in warm rooms and we turn up thermostats and things like that. But if you were a baby, that wouldn't happen. And so, and your surface to body mass ratio is different as a child. So children are more likely to respond to cold with TRH. But then the body recognizes other metabolic symptoms as well. So the point of thyroid hormones is to maintain metabolism and to maintain your basal metabolic rate and to maintain your body temperature. So all of these kinds of detections come into play, and the cells that are going to be releasing this are the paraventricular nucleus and the arcuate nucleus. So this little releasing hormone is only 3 amino acids long, but in order to keep it from being broken down, it has a glutamate that's twisted around and reconfigured, so the long tail of that side chain, and we call that a pyroglutamate on this end, so that N-terminal side gets protected from proteases that can cleave from this end. And then over here on the proline, that carboxylic acid has an amine on it, so that can help keep those proteases that start at the C-terminal side from cleaving this tiny little protein. As it turns out, it doesn't need to have a very long half-life because it's only going through the portal system. It doesn't have to be in the bloodstream. But it can also be found in the brain where it can serve as a neurotransmitter. And then our somatostatin comes from the periventricular nucleus, and that inhibits release. So the pituitary is responding to those signals by making TSH, which is a large protein, alpha and beta subunits. You remember that the alpha subunit is the same thing as the for luteinizing hormone and FSH. It's the beta subunit that has the biological activity, and your immunoassays that detect these are for the beta subunit, so you don't have cross-reactivity. And here our half-life is about 60 minutes, and it's generally degraded. So now our TSH is going to be stimulating the thyroid gland in many ways, growth as well as release and synthesis of the thyroid hormones. So let's look to see what the thyroid hormones are. There are 2 of them, and they're called either T4 or T3 based on the number of iodides that are on these aromatic rings. And if you were just to look at this part without the second aromatic ring, you'd probably say, aha, that looks a lot like tyrosine. And it does, because it is. Tyrosines are going to be bound within the membrane structure, and this side chain here is going to be binding to another tyrosine side chain after these iodides have been added to it. So if you have 2 iodides per aromatic ring, oh, and this is that little hydroxyl at the end of one of those tyrosines. This was the hydroxyl at the end of the tyrosine before you got the covalent bond with the other aromatic ring. So 1, 2, 3, 4, that's T4. Now, if you don't have a lot of supply of iodide in your bloodstream, you may not have enough. And some of your tyrosines only may get 1, and others may get 2. So if you have one that has 1 binding to one that has 2, then that's called triiodothyronine, and that's this. So those are the 2 thyroid hormones that are going to be released in response to TSH. What are their functions? Well, the 2 that you want to remember are they're necessary in a permissive way for growth. So you have these hormones as genomic regulators. The thyroid hormone works by going to the nucleus and stimulating new transcription. So you need to have the enzymes that the thyroid hormones can release in order to have other hormones in the body working using those enzymes and proteins. So you could say it facilitates the effects of other hormones. And then it has a main metabolic rate to increase the body temperature and increase the metabolism of virtually all the cells in the body. And it does this by uncoupling proteins that it can synthesize that can regulate whether the mitochondria are going to be generating heat or whether the mitochondria are going to be generating ATP. So these uncoupling proteins can turn off part of this so that your cells in your body are making heat. Okay, so our little pathway through the hypothalamus, we got our release. If we have somatostatin, it tends to be producing a tonic inhibition. So you need to have this stimulation to be able to get the release of TSH. Then your TSH gets into the bloodstream. It stimulates the thyroid gland. It stimulates the growth of the tissue. It stimulates the blood supply. It triggers the release of thyroid hormones after it has already triggered their synthesis. And so you can see here that once you've got the thyroid hormones in the bloodstream, they're going to target the pituitary as a long feedback mechanism. So the arrows here are two different sizes for a reason. The main regulation of TSH released from the pituitary is the feedback mechanism from the thyroid hormones themselves. And the reason I have a little arrow here going from T4 to T3 is that the body uses enzymes called deiodinases, which are going to take the iodides off. And so when you take that iodide off from T4, then you have T3. So as soon as you get to the thyroid hormones, or the thyroid cells themselves, you find that there's a deiodinase in those cells that are going to be making T3. So the T3 can either come from the serum or it can come from this. And the clue here is T3 is the much more potent, much better hormone at the thyroid hormone receptors. So all these little take-home messages we're going to be talking about at great length in the next 35 minutes. Well, while we've got this diagram up, let's take a look at the thyroid gland itself. So the thyroid gland is made up of what we call follicles. And so this follicle right here is a series of cells that are cuboidal epithelial cells. And when they're working hard, they're going to be nice thick cubes. But when they're not working hard, they're going to be thin. And inside your follicle is what we call colloid. And that colloid is made up of the protein thyroglobulin that has those tyrosines on that are making the thyroid hormones. So this is all a proteinaceous, jelly-like internal structure here. So this is your thyroid follicles. Now, when we talked about the thyroid glands in class the day before yesterday, I told you that parathyroid glands were embedded in here. You don't see any parathyroid glands embedded in here for the parathyroid hormone release. And then there were C cells that were in the thyroid gland that were releasing calcitonin. C for calcitonin. You don't see any of those peppered in here either. All you see is these blood vessels. So the artist who was drawing this didn't care about bone regulation with, so that's why they didn't draw them in here, but that's where they would have been, but they would not have been part of these follicles. Okay, so how are thyroid hormones synthesized and released and transported, and how does TSH work here? Well, we've got our follicles right here. The blood supply is outside the follicles, and that's where you want to be able to get nutritional supplies into these cuboidal epithelial cells so that they can make See, they're not part of the follicles, and they're labeled right here. Okay, and in case you didn't realize it, your thyroid gland is right here in your throat, right along the trachea. So you can actually stare at people on the bus and tell whether they're potentially thyroid deprived and driving their thyroid glands and they've got a goiter, or maybe they might have nodules. So you can just sit around making diagnoses about people that you see in church and bus. You know, this is your life now. You can walk into a room and tell when somebody has had side effects of a schizophrenic drug. Has anybody ever noticed anybody like that? No? Okay, well,
Noticed
because now
schizophrenic.
you've had some pharmacology in your previous, you can begin noticing people, and you should. That's why you're here. So anyway, this is really very noticeable when it changes. So if we look at one of these follicular cells here, this is the blood supply over here. This is the colloid interior here. So we say that this is the apical side of that follicular epithelial cell, and outside here is the basilar side. So this is basilar, this is apical, and it's because if you were to make it look like a pie, the little sharper edges would be here on the inside. So here we have our iodide coming in through a pump, which is on the outside of the cell, and it brings in the iodide, transports it by diffusion throughout the cell, but then it has another transporter that takes it right outside in the apical surface so that it gets into the colloid, and that transporter is called pendrin. So we've got our sodium iodide transporter over here and our pendrin over here. So this is a very, very, very efficient way of getting iodide into thyroid glands. And no other cells in the body, except potentially some of the follicle cells that are in the ovary, have this potential. So once you have a meal that has iodide in it, and you're going to get that from iodized salt, you don't have very much iodide because it's cleared so rapidly and stored here in the colloid. So the other thing that these cells are doing is making the thyroglobulin. So here's a depiction of probably a rough endoplasmic reticulum where this large protein is being made. And then that gets secreted because it's made, it put in a vesicle, and secreted right in here too. So here's our protein. It just shows you a little wedge here to indicate that you've got tyrosines on your protein. And those tyrosines are going to come in contact with the thyroid peroxidase, which is going to make this from its charged form to now an oxidized form, and that way the iodide can bind to the tyrosine while it's still on the thyroglobulin. So let's talk a little bit about this iodide pump first, because this is important. Maybe not in this country, because in this country we very rarely have people who are iodide deficient. But in most countries of the world that do not have iodized salt, and in the United States before about 1935, we did not have iodized salt. And people in the Midwest were all getting goiters. And I'll show you a goiter in a minute, because they were not having any iodide, they could not make any thyroid hormone. But because that feedback mechanism is largely from thyroid hormone itself, if you're not making thyroid hormone, you're making a lot of TSH to be released, and it drives the synthesis of the gland. So this is really a very important kind of pump. So it's a co-transporting pump. Here's your sodium potassium ATPase. And so you've got potassium being driven in, sodium being driven out, and that sodium is now going to drive the little pump that's right next door to it, and this is a sodium iodide symporter. So it needs both the sodium with a positive charge, the iodide with a negative charge. You need two sodiums for every one iodide, and it drives it right in. So sodium comes out here, is pulled right in here. Drives in the iodide with it. Then on the apical side of the cell, you need the pendrin. That is not driven at all by ATP. So the ATP part, the active part is right here. Very active, drives in most of your iodides so that it can trap 50 times perhaps the amount of iodide that would have been in the bloodstream. So this is what happens if you don't have the thyroid hormones being made. You have the TSH driving the vascularity and the size of the gland. So even though she may not be making thyroid hormones, her gland is extremely large. And we call this an endemic goiter because it's not related to any intrinsic endocrine dysfunction. It's just that the endocrine system is properly working to try to increase the size of a gland, but the iodide isn't there to do that. Now, most of us don't have any trouble keeping 500 micrograms per day of iodide in our diets because you have lots of hidden sources of iodide. It's even found in milk, probably because farmers use an iodide-like solution like betadine. Any of you do animal surgeries and have used betadine scrub? So that can get the iodide into milk. But it's also kind of a preservative. So if you look on your package insert on white bread, you'll see that there's iodide in there as a preservative. It's in toothpaste. It's on in a number of places. So in our country, we hardly ever have this problem. But you can see if you bring this in, most of it is driven immediately into the thyroid. And then it's made into our thyroid hormones, which are released. And as the liver and other tissues remove those iodides can get right back into circulation. And then they can be taken back up into the thyroid, or some is released in the stool, but most of it comes out in the urine. So what goes in generally comes out, but in the meantime, it's already been made into thyroid hormone.
Dr. Howard?
Yeah.
For the previous slide, the patient with the goiter,
Yeah.
will just daily treatment with iodine?
Yes. Oh, yes,
slowly
that'll resolve
That's
it.
it? That's all you need to do?
Yes. Now, it won't do it tomorrow or the next day,
Yeah.
but it'll do it within a very short period of time.
Cool.
Or you could simply give her thyroid hormone to help reduce this, and the thyroid hormone will release the pituitary from releasing TSH, and then you could get the iodide back. So, yes, very, very, very treatable, extremely treatable. Okay, now this is our thyroglobulin, 660,000 molecular weight. Now, how many of you generally run SDS page in your lab? And what kind of protein sizes do you generally see on that ladder that you've got?
Like up to 200.
Up to 200. So is this a big protein or what? This is a huge protein compared with most of the proteins that we see in the cell. And of this huge, gigundous protein, there are 123 tyrosines, but only 4 to 8 of them are available to have iodides added to them. Isn't that bizarre? I mean, doesn't that strike you as a waste? Doesn't that make you wonder what God could have been thinking? What on earth is happening here to put all of that energy into 660,000 molecular weight? That's, since amino acid is generally about 100 molecular weight, that's close to 7,000 amino acid steps that you have to add to get that entire thing for only 4 to 8 tyrosines. And you need 2 tyrosines, so that's only 2 to 4 thyroid hormone molecules. That just seems such a waste of energy. And I don't quite understand why that happens. It doesn't make at all sense to me. But but I wasn't part of the human body planning process, so, you know, I can't really speak to this. But if anybody has any interesting ideas about why thyroglobulin would have to be so big, number one, and why so few productive thyroid hormones can be generated from it, you know, it's just unclear to me. So depending on how much iodide you have, if you have low iodide, you get less iodides per molecule. Probably you'll end up with T3. As you increase TSH, you'll have greater T3 levels because you'll be driving the gland to be releasing. So those are two ways in which this ratio of T3 to T4 can be altered. In general, we're going to generally say that in a healthy individual, in a normal environment where they're getting nutritional iodide, maybe 90% is T4 and only 10% is T3. Yeah?
Back to your last comment, could it be because you don't want to prevent the bonding between those tyrosines? I mean, those, tyrosines?
Could be. But that requires an enzyme, so it's thyroid peroxidase. And I think the first job thyroid peroxidase does is to put the iodides on. So, and you actually do want that bonding. So, so let's just say if the first step, if it requires having the iodides on first, then I don't think you're going to have the random bonding.
Or even like covalent bonding between all of them, which would be kind of strong in this iodine?
I don't know that that's going to happen. And I think that those that are available, the 4 to 8, they're on the surface of the protein after it's already folded.
Oh, okay.
So the others are going to be inside, you know, the protein needs to be folded in three-dimensional space, so it's going to be a globular protein. And I don't think that's it. Okay, so once we have built this with our thyroid peroxidase, and the production of thyroid peroxidase is under the influence of TSH. So the synthesizing of pendrin is all under TSH regulation. So once we have it made and it's still bound to the thyroglobulin, when TSH stimulates the release, TSH does it by promoting the endocytosis into the cells. And it does it by little proteins called megalin that begin to initiate the invagination and the internalization, and that the interior of these vesicles is now colloid. And the colloid thyroglobulin has the T3 and T4 bound to it. So then this little endocytic vesicle goes directly to a lysosome where the proteolysis takes place and the T3 and the T4 are released. They're lipid soluble, so they can pass right out of the cell membranes and into the bloodstream. Meanwhile, the diiodotyrosines and the monoiodotyrosines are free to be degraded. So if you're looking at the histology, if you don't have TSH, then you see rather thin, inactive epithelial cells in the follicle. But when you've got good activity, then you see nice thick cells because you've got lots of new protein synthesis going on to keep that thyroglobulin being made. And then you'll be likely, if you look at your histology, to be able to see on the apical side the invagination, where you can begin to see the thyroglobulin being taken into these endocytic vesicles and broken down. So what does TSH do? TSH is growth of the gland, so that's hyperplasia, so lots of mitogenic activity to be able to get new proliferation of new cells in the gland. Increased vascularity, you need to have that blood supply. Synthesis of the thyroid hormones, so it's iodide trapping, as well as the thyroid peroxidase, all induced in response to TSH. Release of thyroid hormones, so the endocytosis, the proteolysis, block these effects of TSH. And that can be very useful therapeutically because if people are hyperthyroid or have thyroid nodules, you want to be able to remove those. You could do that surgically, or you could do it with radioactivity. Either way, a hyperthyroid person, first of all, has a very driven gland, so it's going to have lots of stored colloid, and it's also going to be highly vascularized. So anything you do to try to destroy a hyperthyroid gland is going to have a tremendous amount of thyroid hormone going to be released into the bloodstream, unless you can curtail it a little bit. So what we typically do is treat with a very high level of iodide for a short period of time, let's say 2 weeks or so that you can begin to reduce that vascularity in the gland and reduce some of the storage of colloid that might be in the follicles before you begin to do what would be a more definitive treatment. So this can be taken advantage of. And the effects don't last very long. So after maybe 2 or 3 weeks of high iodide, that effect goes away. Looks like I have too many S's on my X's. Okay, now we've got our diagnostics. So, because you can see iodide getting into the gland using radioactivity, you can provide a radioactive source like I-131 or something that looks like that. So here's a technetium ion with the oxygens on it, makes it pertechnetate. And that's about the same size as I-131. So that's taken directly up into the gland, and the X-rays can pick it up. So you can see that over a course of time, if a person is normal, then the radioactive iodide is going to be taken up into the gland, and you can be able to see it on an X-ray. A person whose thyroid gland is not functioning and cannot take up iodide is going to be less likely to have radioactive iodide taken up. And a hyperthyroid gland or hyperthyroid nodules is going to take up even more. So you can use this as a diagnostic measure. Now let's talk a little bit about transport, metabolism, and binding proteins. And this is very important because thyroid hormones, being lipid soluble, need to have binding proteins. And there are at least 3 different kinds. A very high affinity binding globulin that's specific for thyroid hormones. So it's called thyroxine binding globulin. And there's not very much of that in the blood supply, but on the other hand, it's binding up almost 70% of your T4 and better than half of your T3. And then something that has medium affinity, thyroxine binding prealbumin fraction. There's more of it, but less affinity, so less is bound. And then you've got all the albumin, which is extremely low affinity, but you've got so much of it in the blood supply that it's going to be contributing, and this is low affinity. So you can see that the bulk of the high affinity is taking up T4. There's less affinity for T3, and so less of it is bound. Now because of that, we have a very long half-life of our T4. So the T4 that can be released from the gland is likely to remain in the blood supply for at least a week. T3 is not nearly as high affinity, so that half-life is going to be about a day. And the tissue, the supply of thyroid binding globulin is largely dependent on some of our endocrine effects. So it's inducible, it's made by the liver, and when you are pregnant and you need to have thyroid hormones supplied to maintain growth and metabolism, you need to have the estrogen stimulating the production of new thyroid binding globulin. So typically we might have 99.96% found, and it's only the free So this is in equilibrium, but what essentially that means is, particularly for thyroxine, that we have a very large storage supply in the bloodstream. And once that supply can be released, it can get into the tissues, and it encounters deiodinases. So the first one that it encounters is a deiodinase in most of the tissues. So there's several kinds, deiodinase 1 and 2. And it's going to be removing the iodide from this outer ring. And that makes T3. So even though the bulk of what was released from our thyroid gland is T4, when it gets to the tissues, it's going to be made into T3. And T3 works better at its receptor in cells. So for that reason, we could call T4 a pro-hormone, because your cells are going to be making it into T3, which is the better hormone. So you can see that even though we don't have very much released from the thyroid gland compared to T4, the bulk of our T4 is going to be made into T3 while it's being metabolized at the tissues. Now there's another one in which deiodinase 1 or deiodinase 3 remove the iodide from the inner ring instead. And you might say, oh my goodness, that's a mistake. Because what it actually does is make an inactive compound. Well, so that's how the body inactivates thyroid hormone, and that's fine, but you might ask the question, why is the body making so much of this? It's called reverse T3, but it's not very active. And in fact, you can see that there are conditions in the body in which the body is making more deiodinase 3, so that it's actually making more reverse T3, going through all of that trouble to make the T4 and then reducing it. Well, before we answer that question, maybe you can think about it. The other way that the body gets rid of its thyroxine and T3 is to conjugate it with sulfites and glucuronides so that it can come out in the urine. But it can also be recycled. So it can go through the bile recycled so that either it goes out in the feces or can come back into the tissue. So we can do a lot to conserve that iodide. So does anybody have a good idea, now that I'm showing you this slide, why the body might want to make reverse T3, which isn't really very active? Well, what's the point of making thyroid hormone in the first place?
So if it's freeing up fats, would the reverse T3 then be storage of fat? Because it's
It
active?
could be,
And
yeah.
this decline in metabolic rate that you've got here?
Okay.
So
Now,
would you-
thyroxine's not your main fat regulator. Growth hormone was.
Oh,
But what does thyroxine do? It maintains your metabolic rate. And in a minute, we'll see some of the other things that it does. Supposing you're sick, do you want your metabolic rate to be driving the body when you're supposed to be staying in bed getting better? No, not necessarily. What about if you're starving? Or you have very poor nutritional state? Then you probably want to conserve your energy. So you can see here that in this particular case, which is a situation of starvation, over a period of a week of food deprivation, now you can see your thyroid T3 levels are plummeting, your T4 levels are being reduced. Your T3 levels are plummeting because they're made from the T4. And so if you are now using the deiodinase 3 rather than the deiodinase 2, now you're making reverse T3 in abundance and less T3. So over the course of a week of starvation, now you have reduced your metabolic use. So you're not driving the body's metabolic activity at a state when you don't have the energy stores and the nutrition to maintain that. So if you can't eat, you don't want to be driving away all of your energy stores. So the body has a natural way of doing that. If you're sick, the same thing. So if you have liver disease, that is probably a very similar situation. So these are the kinds of things. Selenium deficiency, you need selenium as a cofactor in a lot of enzymes, and particularly those that can protect you from oxidative stress. Oxidative stress is when your mitochondria are working really, really hard. So a selenium deficiency, it's important to make sure you don't get oxidative stress to damage the tissues. And so the body's result for there is to decrease the metabolism as well. So I think there's a reason for that. Except now we've got to worry about our pituitary. So our pituitary can be generally inhibited, the D1 iodinase, by our calorie restriction or our stress. But the deiodinase we have in the pituitary is refractory to that because it's a different deiodinase. So you can have all those feedback mechanisms going into play because the pituitary uses a deiodinase 2 rather than deiodinase 1 or 3. Okay, now, what are the main actions? And I told you at the very beginning it was going to be metabolism and helping body growth. But let's see how that works. Our thyroid hormones are brought into the cell passively because they're lipid soluble. And among the first enzymes that they see here is a deiodinase. So that's breaking down our T4 to T3 as it gets into the cell, as somewhere in the cell before it can cross the nuclear pore to get into the chromatin. Now its receptor is already there, bound to the chromatin, and it has a partner because whenever you have transcription factors, you generally have a dimer. And it's not a homer dimer. It's generally binding to retinoid X receptors, so it's a heterodimer. And when you don't have thyroid hormone, it serves as a repressor. So it actually keeps transcription from taking place on those key genes that it's going to regulate. It's only when you can get the T3 into the nucleus that this now becomes active and you can get your gene transcription. So we have several nuclear receptors. They're going to be alphas or betas. We have a series that are going to be in the heart, and those are alpha 1. There's an alpha 2, but it doesn't work through thyroid hormone receptors. It seems to be very homologous to the viral protein called CERB-A. So you've got viruses that seem to have hijacked some of this, but they don't seem to function here in the regulation of thyroid hormones. Then we have betas in the brain, kidney, and liver, and the beta 2 is in the pituitary. So again, the pituitary and the hypothalamus have a different kind of thyroid hormone receptor subtype than is in other tissues of the body. So this one is kind of protected, so we can always have feedback mechanisms even if we don't have functioning in other tissues of the body. So let's look and see what some of the effects are. We already talked about growth. So our thyroid hormones are permissive so that growth hormone can work, cortisol can work, and particularly we can get bone growth. Our metabolism, our food intake, mobilization of fats and carbohydrates, thermogenesis, that's building our body, heat production, sweating, all under that regulation. In the synapse, we have developing proteins that are necessary for building synapses, myelination, and cell migration is all very important, even on into adulthood. Cardiovascular tissue, we have increased rate and force of contraction in the heart, and one reason for that is that the heart produces beta adrenergic receptors so that you can respond better to epinephrine. So a little tiny bit of epinephrine can do a very good job of increased heart contraction, simply because you've got more beta-adrenergic receptors, the more thyroid hormone you can stimulate. And then all these metabolic enzymes, increased mitochondrial production, increased sodium potassium ATPase, increased oxygen consumption, increased glucose absorption, increased gluconeogenesis. These are all things we're going to talk about next time. But all of that is under thyroid hormone. So our metabolic effects, here's our basal metabolic rate. You give a little thyroid hormone, you get a lot of activity. And our basal metabolic rate, the more you give, the more you get. And this is largely because the introduction of uncoupling proteins, UCP2, UCP3, in skeletal muscle. So most of your heat production is going to be your skeletal muscle. And that's where these uncoupling proteins can go into the mitochondria, block the production of ATP, but are driving mitochondrial production, so that drives heat production. In our metabolic rates, we need to have carbohydrates, so we increase the action of epinephrine and all of the tissues by increasing beta-adrenergic receptors. So you can see we're highly interactive with other tissues. The goal is to increase oxygen production, heat production, body temperature, and the utilization. And we're stimulating in our bone growth. So you can take a look at this. We'll be talking about some of these metabolic effects next time, so I won't go into a whole great detail here, but you can see that the thyroid hormones are involved in virtually every tissue of the body. So if we have too much, we've increased our basal metabolic rate. This depiction of a patient has very decreased body weight. You can see her bones are just sticking right through here because her metabolism has gone much faster than her food intake. And so she's nervous, hyperactive reflexes, heat intolerance. She's building heat when she doesn't really need to. So if you asked her which part of North Carolina she likes most, she's going to say the cold in the winter. And muscle weakness, because she's driving her weakness. She's driving smooth muscle as well, so diarrhea. Increased appetite, and I didn't spell that correctly, did I? So you can see all of this, and obviously she's very tired because of all of this. Now the other one, this is why my friend's wife rushed him to the emergency room because he was acting stupid. Here we've got a decreased metabolic rate, cold intolerance. So this lady's going to love it in the summertime. She needs to have the body heat because she's not making it herself. Interactions with other tissues of the body that are involved in endocrine responses. So she may not be cycling very well in her in her menstrual cycles. Goiter, look at that. The goiter's not, or the gland isn't working, so TSH is driving it. Weakness, fatigue, lethargy, sweating, some other things that I'm not exactly sure exactly why you get hoarseness and thick tongue. Psychosis, all of these. And if it takes place during development, here are some fraternal twins. This one is normal thyroid. This one was thyroid deprived during development. And you can see that both of these are 8 years old, but he's at the level of a 3-year-old, mentally as well as physically. So I want you to take a look. The rest of these slides are going to be showing you a little bit about this feedback mechanism that we've harped on over and over again. But in answer to your question, here we have a diagram that's showing us the response that cells make in response to a little injection of TRH in terms of releasing TSH. And you can see that at very low thyroid hormone levels, you get a big bolus of TSH being released. But over time, 24 hours, 48 hours, as you provide thyroid hormone, the response in terms of releasing TSH is reduced. So this is that feedback mechanism. The more thyroid hormone you got into the tissue, the less likely the pituitary is going to be able to respond to release the TSH. So take a look at these two diagrams, because these are what show you why it's so much better to use TSH as a diagnostic tool as to whether you're functioning well as a thyroid gland, as a pituitary, or as a hypothalamus. So you can begin to identify what happens over a period of time, whether you've got high thyroxine and it's suppressing TSH production, or whether you have limited thyroxine and you have a high TSH production, and whether this is appropriately related to a problem in the hypothalamus, the pituitary, or the thyroid gland itself. And so try to think through the logic of all of this as you begin to think. You can measure TSH very well in the bloodstream. Okay, so thank you very much for coming today, and we'll see you again bright and early on whatever that next day is that we meet. I think Dr. Eldridge is talking to you on Monday. Right? Yeah. All right. And that'll be here. So anybody who's getting confused about where we are, please